So thanks for joining the third week of the stem cell training program. And then now we have over 220 registered participants. For those who've joined recently, and just a brief introduction, I'm Ki-Tai Kim, Director of the UCLA Human Stem Cell and Genome Engineering Center. And our center is focused both on the collaborative research and also core service in iPSC programming and gene editing led by Dr. Hyo-Kyung Cha and Dr. Dennis Atta. And some of the participant institution can access to our center service through the institutional partnership that will happen this year. So therefore, I will notify and then you will recognize our center within your institution. And this program is supported by California Institute for Regenerative Medicine in collaboration with Thermo Fisher Scientific. And as a reminder, participants who attend at least 6 lectures will receive a certificate of completion training. So please make sure when you log in with your full name for attendance tracking. So if you log in with your partial name, and then please write your name and the email address in the chat box. If you attend without registration, please register following the information in the chat box. I will post the registration link. And if you already missed 2 lectures, and then don't disappoint, and it is still worth to attend to get the certificate by attending the mixed lecture next year. So therefore, I continue to attending the class. And today's lecture will cover the basic feeder-free human pluripotent stem cell culture, including key principle, material, and then practical consideration. And we also pleased to have a special guest speaker, Dr. Ming-Fan Ho from Mayo Clinic, and who will present on stem cell clinical application. And she is famous with a mini brain organoid system. So now I would like to hand over to Omar and Tom.
Thanks, Kitai. All right. Can everyone see that okay? And can everybody hear me okay?
All good, Tom.
Yes.
Thanks, Omar. Oh, well, thanks everyone again for joining us. We're really glad you're here. We do have a pretty packed agenda today, so I will get cracking in the interest of time. But as Kitai mentioned, our goal really today in the first part of the lecture is to introduce some basic features of feeder-free PSC culture, right? And then as Kitai mentioned, we've got a really important special guest talking today about their research, and that's Dr. Min-Fen Ho from the Mayo Clinic. I had a quick look. She's done some really, really interesting work, everything from multiomics and drug response in substance use disorders. She's also done some really cool stuff with hPSCs, generating mini brains in a dish. So really excited to hearing what she has to talking about and hearing about some of her work. So as always, though, as we progress through the lecture, feel free to chime in with any questions. We're always happy to take them, and you can certainly reach out to us afterwards as well. Although if you send us an email afterwards, it might take us a few days or so to respond, but we will get back to you, promise. So just to be a bit more specific about what we're going to be talking about today, as I alluded to, I'm going to give a quick overview of some of the content that we've covered so far, which we always like to do just to set the stage a bit. I'm going to touch again quickly on how we define stem cells, especially in the context of disease model generation. And then going to switch gears a bit and touch on a couple of different cell culture formats, right? In our case, namely adherent, suspension, and 3D organoid culture. And I'll mention a few important considerations surrounding each. I'll then segue into PSC culture systems, where, as Kitai mentioned, we'll define sort of feeder-dependent and feeder-free systems and talk about the importance of things like matrices, substrates, media system considerations, passaging methods, right? And we'll get sort of set the stage today, and we'll get into a bit more detail on some of that stuff next time around when Omar does feeder-free advanced. And then we'll also talk a little bit about cell culture challenges. Cell culture is not easy, which is why we take courses like this, and we try to always improve upon our cell culture game, right? And it'd be cool if some of you would like to share some of your experiences or challenges that you've had in the lab, and maybe even how you've managed to overcome them. So when we get to that section, maybe you can pop some of those in the chat. I think it's always interesting to hear what other folks have done or have gone through, and we've all got stories for sure. I then close out quickly at the end of the first part of today's session with a very brief case study talking about hPSCs in the context of immunology. And then, as I mentioned, I'll hand things over to Dr. Ho. Okay? All right, well, let's get cracking. Oh, and I put this in there. I think this is a really cool picture just before the quick review. Anyone want to take a guess what this picture resembles? And this is not Omar waking up in the morning either, by the way. Pop it in the chat if you think
I
you
was
might.
gonna say it looks pretty close, Tom, to me waking up in the morning.
These are what's called neural rosettes.
Nice.
These are one of the primary developmental signatures of neuroprogenitors in culture. These particular ones were derived from human embryonic stem cells. You can kind of see how these rosettes have this sort of radial configuration, which is pretty typical. So they kind of diverge out from a common center of, in this case, columnar cells. We're kind of looking at maturation unfolding here. You can probably guess in the eye, and using a neural marker called PAC6. Where it works in collaboration, we know, with another marker that you may have heard of if you work in neurobiology called SOX2, which is a pretty well-known marker for ESCs and neural stem cells. But pretty cool image, I think. I'm always fascinated with the image side of things, and I think it can certainly tell a story for visual learners like myself when you look at a pretty picture like this. So anyway, last week, Omar took us down a reprogramming journey, if you will. He talked a little bit about the advent of iPSC technology, specifically in the context of somatic cell reprogramming to generate our beloved iPSCs, right? Our induced pluripotent stem cells. But again, we defined what exactly stem cells were and what we mean when we throw that term around. So in the most basic sense, right? Stem cells abide by two primary rules. They can self-replicate, as it needs to make more of themselves. And second, these are cells that can differentiate into other cell types. And there's a number of different types of stem cells, right? So we talked about stem cells that have kind of this unlimited capacity, if you will, for self-renewal. There's PSCs like embryonic stem cells that we talked about derived from a blastocyst, and also induced pluripotent stem cells that are generated from typically reprogrammed somatic cells, which is what Omar talked about last time. Both of these types, ESCs and iPSCs, again, can become just about any cell type. But then we also have cells with a much more limited renewal capacity, right? Like these adult multipotent stem cells on the right here. Again, a couple of just important distinctions here are that these cells can renew themselves a number of times, these adult multipotent cells, but again, by no means indefinitely. They typically differentiate into only certain cell types, right? Typically the tissue from which they were isolated, as you can kind of appreciate on the right here in this table. Now, as we alluded to initially, right, we're capable of taking our blood or skin cells, for instance, and as Omar mentioned last time, forcing the expression of certain protein transcription factors to actually reprogram their cells into iPSCs. Again, with that technology, we can now edit them to introduce mutations and then differentiate them into a number of different highly functional cell types, right? Think cardiomyocytes, neurons, immune cells, which we'll talk briefly on today. And then from all that, right, generating these pretty incredible models and a whole bunch of different downstream applications. And again, these are some important considerations on the right here, right? We talked about patient comfort, sample size, the ability or experience needed to maybe reprogram even a certain cell type, the method that you're going to use, right? And then, of course, you have to think about things like intended downstream application. And again, we show this slide a lot. You've seen a rendition of this before, but I think it puts things into context to highlight really what the stem cell disease modeling workflow looks like from a high level, if you will, and how these different individual components or buckets, right? Cell models, reprogramming, engineering, characterization, differentiation, different forms of downstream analysis, right? They allow us to create these physiologically relevant models from which we can study a whole bunch of different disorders. And we talked about this in the first lecture in the context of Parkinson's disease, right? When we generated dopaminergic neurons. And the case study I chose for today, just a quick one, is one that actually looks at the use of engineered iPSCs for hypoimmunogenic studies. So hypoimmunogenic derivatives of iPSCs that are actually able to evade immune rejection in recipients. So pretty cool study. And again, just to drive home kind of big picture here, there's lots of promise, as you know, underlying the basics of stem cells, right? We've got basic biology where we can understand maybe things like how to treat birth defects or neurodevelopmental disorders, which is where I've done a lot of my work. Drug discovery and development. This is a cool one, right? We can use stem cells to test the effects of experimental drugs on actual patient cells. I believe our guest lecturer does some of that work today. Disease modeling, right? Sky's the limit here. There's loads of different nowadays patient-specific or disease-relevant cell types that allow us to understand human disorders. And then even taking that one step further, right, in the context of cell therapy or regenerative medicine, where we actually harness or take some of those inherent capabilities of stem cells to almost ideally act as a repair kit, if you will, right? So there's loads and loads of potential surrounding stem cells. And as you'll hear Omar and I talk about throughout this course, I really think we're only at the tip of the iceberg into, let's say, tapping into and really impacting how these different applications of stem cells affect modern medicine. So let's segue into a little bit of some of the basic science and a few of the mechanisms underlying all this stem cell potential, and touch on a couple of different cell culture formats that you might encounter in a lab, right? And again, we start here by first defining cell culture, right? So cell culture is really the process by which cells are grown under controlled conditions, right? And generally outside of their natural environment. After we take those cells of interest and isolate them from, say, a living tissue, or maybe even reprogram them into pluripotent stem cells, as you know, they can be maintained under very controlled experimental parameters, right? Where we use them, a good way to think of this is almost as a means to an end, right? But not always necessarily the final product. So a nice analogy that I put on the top right here is to think of cell culture almost like a connecting flight, right? In that they typically aren't the final destination maybe of our research, but a tool that we're using along the way to almost study and improve upon different applications that we just talked about. And along this journey, if you will, if you go back to the flight analogy, you have to feed the cells regularly, right? We have to observe the cells for important signs of health, like their morphology. We have to subculture or passage the cells, right? In which we remove spent medium and then transfer the cells from, say, a previous culture dish into fresh growth medium, which again further enables the further propagation of these cell lines. And nowadays, there's lots of different formats that enable this underlying process of cell culture, each of which are slightly different and all have certainly their own merits. Adherent culture, which is probably where I would say most people's cell culture experience lies. There's also suspension culture. We're seeing a lot more of this nowadays, especially as a means to generate a large number of cells that really, due to sort of their shape and inherent properties, are more physiologically relevant, say, adherent culture. There's also other formats, right? Like 3D formats, like 3D organoids. These are really cool little models, and we'll talk briefly on them today and throughout the course as well. These even more closely resemble in vivo cell environments, and I think are really all useful models from which we can work to, again, help us ultimately understand human disorders. Now, I'd say more traditional cell culture systems like adherent workflows, what those do, if you think of it, is they grow cells on a flat, coated plastic dish, right, in which the cells are then dissociated, maybe enzymatically, for instance. And that works quite well for a lot of different applications and is, maybe appropriate for most cell types, we could say. And that's, nowadays anyway, partly because adherent culture is quite easy to work with. It's quite well understood as a means to culture stem cells. But you've got to think, right, these systems are not necessarily representative of what actually occurs in the human body, right? They require some form of tissue culture treated vessel, right, which allows for cell adhesion and spreading to occur. And so I think one important consideration or point of distinction to think about with adherent culture is that growth in adherent culture is often limited by the surface area of your particular culture vessel, right? Meaning it takes a lot more time and often resources to, say, generate a large number of cells, which is, we need a large number of cells, right, for things like preclinical research, drug screens, and a lot of models nowadays that for one reason or another require a lot of cells. And so nowadays, as I said, many cell lines can also be adapted to spheroids and, say, suspension culture, Suspension culture is quite easy to work with nowadays. We can maintain those cells with a bit of agitation, use that agitation to encourage things like gas exchange, typically in vessels that are not tissue culture treated. And so, generally speaking, suspension culture gives us, I would say, a more predictive physiological response than, say, adherent or monolayer culture. And again, this is due to a lot of the complex and sort of multicellular nature of spheroids in suspension that mimics that in vivo biology a bit better, and also allows us to generate a heck of a lot more cells in quite a short amount of time, especially compared to, say, adherent culture. So as I have on the right here in this nice image, suspension culture is in some ways a nice bridge between, say, the research bench and the clinic, because it gives us a closer resemblance to how organs, for instance, fundamentally form and actually grow in the human body. So along those lines, if we put that sort of notion into a clinical context here, I'm using an example here of looking at the expansion capabilities of suspension culture versus adherent culture, and kind of highlighting a couple of differences here in things like hands-on time involved, potential pain points, and sort of a loose estimation as to the overall cost of each workflow. We use an example here of generating a whopping number of 4.5 billion cells. And if we were to take this in context of preclinical research, this would be about the number of cells that you need to treat 2 patients with a cardiomyocyte cell therapy. You can see along the bottom here that with maybe a traditional monolayer culture or adherent workflow, and starting on the left with 1 well of a 6-well plate here, it takes us about 28 days or so and about 6 different culture formats to scale up and ultimately generate our 4.5 billion cells. By comparison, though, if you look across the top row here, and in this case, utilizing suspension culture, it takes, I would say, considerably less time, about 19, 20 days, as well as less culture formats to create that same number of cells. Now, think of this too, that in addition to lowering the chance of contamination in suspension culture, right, by reducing the hands-on time, expanding PSCs in suspension often uses less media as well to attain the same number of cells, in many cases making up to quite a few times less expensive. So I want you to understand some of the merits here of suspension culture, but also don't hear what I'm not saying here. Adherent culture really is great. Don't get me wrong on that. But there are considerable benefits when utilizing suspension culture systems, especially if you're aiming to scale up and generate a large number of cells. And then finally, the last of the cell culture formats that I want to touch on today is 3D culture. And again, we'll expand on this a little bit. And yes, suspension culture is certainly a type of 3D cell culture, especially when those cells are allowed to aggregate and form those spheroids in liquid medium, right? Rather than, again, being attached to a surface in adherent culture. We could certainly talk about maybe even spending an entire lecture on the different types of 3D culture models, right? There's loads of them out there nowadays, but for the sake of today's talk, I did just want to briefly mention organoids. Those are my favorite ones. I think organoids are really cool and really useful little models. They're basically 3D structures derived from either PSCs, pluripotent stem cells, or in some cases, organ progenitors from tissue-specific stem cells, in which cells will typically spontaneously organize due to their sort of inherent polarity into differentiated functional cell types and progenitors that in many ways resemble their in vivo counterpart, and therefore, as the name implies, mimic some of their functionality of a particular organ, right? The organoid, right? So they're even more complex in vivo-like than, say, spheroids in suspension culture, giving them a nice microenvironment for very targeted cell research, cell therapies, focused disease models, and so on and so forth. And again, we won't go into these too much today, but very much know that they exist. I think they're a really fascinating little model and one that's incredibly relevant, incredibly useful as far as cell culture formats go. Now, the other thing I want to mention that's also an important consideration is cell culture vessel types, right? And there's loads and loads of different cell culture vessel types nowadays, as you can see here, or I'm sort of alluding to on this slide. And again, we're not going to go into all of these in the interest of time, but I've listed some cell culture vessels here in the context of adherent versus suspension culture. When you're choosing the right cell culture format, you have to plan what you're going to do experimentally, both in the short term and long term. And obviously, before you start your experiments, you want to choose a cell culture format that is good for cell culture consistency, that's good for cell health, that's good for reproducibility of the experiments that you're going to be performing. There's also lots of different types of plastics that we've been using over the years, right, to perform cell culture and that make up these culture vessels oftentimes. I'd say the most commonly used nowadays is probably polystyrene or PS. They're pretty low costs. They have sort of this inert chemistry in them, which makes them, I would say, a good choice for disposable cell culture. Cell culture grade plastics are typically chemically treated with the treated culture vessel surface typically carrying a positive charge. And if we go back to our first lecture, right, you remember that proteins on cell membranes are mostly negatively charged, right, which is why cells are quite easily attached to cell culture grade plastics. You can obviously optimize these vessels by coating the surface various different ways. There's lots of different flavors here. We can use different peptides like polydilysine or PDL, different proteins like collagen, even different polysaccharides are sometimes in use as well. So there's lots of different shapes and sizes and different chemical configurations of these cell culture vessels. Really all just things to keep in mind when planning an experiment. So let's segue a little bit now into a couple of more specific details surrounding cell culture systems. And I want to highlight and really touch on some of the more essential elements of growing PSCs in culture, right? Especially as highlighting the merits of feeder-free culture, right? So we have to think about things like the matrix or substrate, which again is what those cells attach to. It's really important for facilitating growth. The media system that we're using, I would say the media is perhaps the most important component of the culture environment. We'll touch on more this a little bit later on, but media provides you all the necessary nutrients, the growth factors, the hormones that you need for cell growth. It also helps regulate things, right, like pH and the osmotic pressure of the culture. So media is super important. You also have to think about passaging method, right? The process of transferring your cells into a new dish. Again, that enables further propagation of your cell line, making sure that you not only have enough cells for your experiments, but also that you're maintaining optimal health and integrity of the cells that you're actually working with. And I'm sure you've heard some of these terminology before, but there's a couple of different terms that we often throw around in discussions of cell culture. Cell culturing, for instance, or the passaging, splitting of cells, as I mentioned on the last slides, is really important. It's something that's regularly undertaking. It's like a housekeeping task, if you will, to divide and transfer your cells into a new culture vessel to give them that necessary or adequate space, as well as all the necessary nutrients that they need to grow and to thrive. Confluency is also another important term that I'm sure many of you are familiar with. This is one of the primary measures that we know of when to act, right? When to actually perform this subculturing task, which again, you'll do quite regularly. Confluency is an estimate of the percentage of the cell culture vessel surface that's covered by your cells when you're looking, say, under a microscope, for example. So 50% confluency means about half of the cell culture dish or flask is covered in cells. Not to get too much into the weeds here, but we do use a slightly different terminology in suspension culture. And again, we'll unpack some of that later, but instead of confluency in suspension culture, we typically use the term density to describe the number of cells per milliliter in that suspension vessel. So just a quick exercise here, just to estimate the confluency. Anyone have an idea in the top left here? You can throw it into the chat. What the confluency of this box in the top left might be.
I always love these because it varies so much. Right, so in the
Yeah.
chat, you have folks saying 80, 95, 80, 90 to 100, 65 to 70, 30, 80. It's very interesting.
It is very interesting. I would say, based on what we typically see and what we typically suggest, this is about 80 to 90% confluency. In number 2 here, I'd say about maybe 50 to 60% confluency, maybe a bit less down here in 3. What about in number 4? Anyone have or want to take a shot at what number 4? If we're calling this 80 to 90 up here, what this might be down here in number 4?
And most folks are saying 10 to 20.
Yeah, I would say 10 to 20 is a very fair estimate. Nice one. Thanks for hopping in on that.
Tom, before you move on, maybe just a quick point I can make here. And this is in reference to something that also Kitai mentioned last lecture, right? About using certain tools or new automation platforms in order to help streamline some of this, right? This is an important consideration that Tom will touch on in just a second of when to figure out when to passage or subculture your cells, right? Especially for pluripotent stem cells, it's very important because these cells need to be in a very steady or robust environment to maintain their pluripotency status, right? So in this case, there are certain microscopes that are now able to estimate confluency. There's also automated platforms and AI tools that are being built to help with this a little bit, so that it's a little bit more streamlined, because it does really vary between one observer to the other, as you guys can see in the chat.
Yeah, also just a brief mention, some official automation team just arrived in my laboratory, and then to update all of the software. That is including something like automated microscope and the automated this confluence check routinely, even just 24 hours, 7 days a week, including 2 a.m. on Saturday. So that
Well,
kind of tool is kind of amazing tool. Maybe next year, I would better invite them to give some guest lecture. That could be really wonderful practice. Okay,
Yeah,
that's it.
we can definitely bake that in.
yeah, I like it. All right, so a bit more terminology here. We're also really interested, for obvious reason, in the number of cells per given volume that are actually alive, right? In other words, the number of viable cells. We typically measure this with a device called a hemocytometer. I'm sure you've seen these around the lab. This is basically a small glass chamber. I always think it resembles kind of an overly thick microscope slide. And we use this for determining the number of viable cells that are given as a percentage of the number of cells per volume. A lot of protocols nowadays that you'll see will also specify a target viability range, especially at certain crossroads in the workflow, just to help keep you on track. It's a nice point of reference to make sure that your cells are healthy and you're following the protocol as you should be. I'll also include at the end of this slide, by the way, a couple of sample calculations with the deck that we share with you guys, just to have extra resources on hand. Another important piece of terminology is colonies, right? Stem cells, as Omar has mentioned, are often cultured in colonies. These are clusters of cells growing closely together in a dish. Remember, Omar always harps on about this. Stem cells like to be together, and this close contact with each other actually helps them to survive. And then lastly, we sometimes hear the term plaques, right? And these are basically holes in layers of cells that can be caused by things like a virus, and they would typically signify distinct zones of maybe cell death or lysis, right? Almost like these apoptotic areas within the colony. In some cases, what can unfortunately happen is the virus can replicate, it can spread, and these regions of cell destruction or lysis can almost sometimes become visible to the naked eye. So with a few different terms under our belt, let's talk a little bit more about some of those essential components that we need to grow PSCs, which I've listed here on this slide. And before we dive into those a tiny bit here, I think it's important, again, like a lot of the things we do in trying to tell a story, and a story that's successful for you in the lab, keep in mind that successful culturing of pluripotent stem cells requires almost like a very compatible combination of these different elements, right? Things like media, matrices, passaging methods, right? All of which, as best as you can, need to be in sync with one another to, again, best support your cell health factors like pluripotency. So giving a little bit of a historical perspective here. Yeah, please, Omar.
Sorry, Tom, do you mind if I mention one more thing as well on
I'd
the
love it.
previous slide? I just wanted to stress out kind of the point that you made around having the right combination of these elements, right? And specifically for stem cells, this is particularly important because these are a pluripotent stem cells, at least. They're a transitional population of cells in vivo, right? And what that means really is that in the growing embryo, right, where these cells normally are found, right, in vivo, they don't last, right? They're a transitioning cell type that they differentiate into all of your other cell types that make up the embryo proper, right? As Tom mentioned in the first lecture. So the right combination of these elements, right, having the right media, the right matrix, employing the right passaging methodology or method is important to maintaining those cells in that state. Otherwise, you'll find that I'm sure Tom, you'll talk about this, they differentiate or potentially die as well.
Cool. Thanks for chiming in. Okay, so a bit of a historical perspective here. ESCs, human embryonic stem cells, were first derived by a very famous scientist named Jamie Thompson in the late 90s. And he did this by culturing cells on these mitotically inactivated MEFs, mouse embryonic fibroblasts, right? In a medium that was supplemented at the time with FBS, or fetal bovine serum. And so these fibroblasts or feeder cells, what they do is they, in feeder culture, right, provide the different growth factors, extracellular matrix proteins, right? But they're not actually dividing themselves. They help by secreting things like growth factors and different cytokines, which of course help to enable different crucial cellular functions, right? That we're always talking about, pluripotency, proliferation, etc. Essentially serving then as a support system or feeder, as the name implies, for our pluripotent stem cells. So Jamie Thompson's work was really, really important pioneering work, very, very much laid a lot of the foundation for the development of a lot of the PSC culture systems that are in use today. They've changed it over time, though, a little bit to help reduce the risk of contamination from some of the proteins from different organisms they were using, other sort of adventitious agents, if you will. By replacing these MEFs, these mouse embryonic fibroblasts, oftentimes with more human-derived alternatives, right? HFFs, for instance, or human foreskin fibroblasts. They also changed the media a little bit too, right? They replaced fecal bovine serum or FBS with things like knockout serum replacements. This again was done to help reduce the use of different animal components and ultimately to cut down on a lot of the variability here. Again, I think just as a quick aside here, a lot of the evolution of these PSC media systems that we work with today, right, are sort of driven by an interest, if you will, in generating stem cells and again, stem cell models that are useful for applications, right? And oftentimes that means that they have to be almost devoid of non-human or animal. They want to be devoid of non-human animal material, right? Making them what's called xeno-free. And this, I think, again, makes sense in the grand scheme of things, where we strive to correlate, as I said, most of the work that we do, as selfish as it might sound, back to these physiologically relevant human models. And so, kind of in the context of these feeder-dependent cultures, preparation and the quality of the feeder cells that you're using is very critical. As I kind of alluded to a second ago, isolation of the different fibroblasts can sometimes lead to contaminants like mycoplasmas, which you might have heard of. Inactivation or sometimes incomplete activation of the cell cycle can allow these MEFs to overgrow. When that happens, those cells will sometimes totally outcompete the slower growing pluripotent stem cells. And so you have to be quite careful when using these types of systems. Proper maintenance is really, really important. They typically need daily immediate changes, daily inspections to make sure that things like the culture morphology, the general health and confluency of the cells looks like it should. What we do with MEF cells, though, these feeder cells, is typically mitotically inactivate them, as I said, before we use them for culture. This is often done a couple of different ways, one of which is mitomycin C. We also sometimes use radiation to help arrest the cell cycle. And so really the take home here is that feeder-dependent culture relies on these feeder cells to provide a lot of the different proteins, the different growth factors, extracellular matrices, etc., all of which are necessary for those PSCs to grow in culture. And again, these are still quite widely used today. Eventually, though, what happened is different media and matrices were developed, of course, to support PSCs completely independent of those feeder cells. And that was a big concept at the time. These feeder-free cultures, right, they work on sort of omitting that first principle I talked about of those feeder cells and supplementing or developing media, right, that contains all the necessary culture components. Right, again, compensating for things like nutrients, growth factors, extracellular matrix components that might otherwise be missing. And so kind of coming full circle back in 2011, we had some really, really cool contributions coming out of the Thompson lab, once again, who basically published and defined a feeder-free culture system that supported the growth of PSCs without any animal components. This was actually a media that we adopted at Thermo Fisher called Essential 8 Medium. And again, circling back as well to xeno-free products, I think it's important and it's safe to say that in research use only or an RUO setting, not all applications require these xeno-free or sometimes these animal-free origin media, right? some of the newer, more advanced PSC applications actually do benefit in feeder-free culture, especially from a more complex medium, one that might be more optimized, for instance, to support different challenging aspects of the workflow. Think about things like gene editing, single-cell passaging, right? Which led also to the development of other media systems like StemFlex Medium, which is from Thermo Fisher. This is another media system that we have that's geared towards performance and maintenance of stem cells, especially in those advanced applications. And again, despite all the stress that these cells are under, doing our best to keep them happy and healthy. Then we'll talk a little bit about media considerations in the next couple of slides here. Down at the bottom, I've included a couple of pictures of pluripotent stem cells grown under these feeder-free conditions. On the left here, we're looking at human episomal hPSCs grown on Geltrex in Essential 8. And then on the right here, we've got cells grown under the same conditions as on the left, but at 5x magnification instead of 10x. You can see that part of the colony here is intact, while another part sort of contains these flatter, more, I would say, loosely arranged cells, which can be indicative of differentiation. So again, if we really backpedal a little bit here and can big picture consider some of the requirements of growing pluripotent stem cells, culturing those cells on flat plasticware, right, it results in kind of this artificial or two-dimensional sheet of cells, right? And I think we can all agree that normal cells in the body, they experience much more of a three-dimensional environment, right, in which they're very much surrounded by other cells, by membranes, by different fibrous layers, adhesion proteins, etc. And so it's important in adherent culture to provide those cells with things like extracellular matrices or substrates, right, different scaffolds and different proteins that allow us to, again, at least more closely mimic that 3D sort of in vivo-like morphology and give us a much more physiologically relevant environment in which we're really trying to enable those more realistic intracellular interactions. And so in feeder-dependent culture, we often rely at the top here, as you see, on human or murine fibroblasts to serve as our matrix, right? And that's because these are radiated cells. Again, they secrete a lot of the different growth factors or cytokines that we need to support PSCs. In feeder-free culture, though, it's important that we select the appropriate matrix. And there's a lot of different ones out there nowadays. The matrix, I think, is another really important determinant for success, right? We've got things like Geltrex, for instance. This is a soluble form of basement membrane. It's extracted from urine tumors. We typically use Geltrex for routine culture. We also have different ones called like vitronectrin. This is a xeno-free recombinant human protein that was also actually coincidentally designed in the lab of Jamie Thompson. Works especially well with Essential 8. We've also got things like laminins, right? Laminin 521, for example, here. This is another defined matrix that supports feeder-free culture. Laminin, just to give a bit of science behind this, is something that's actually present in blastocysts. And so it provides PSCs an environment that very much mimics the biology, if you will, of the human stem cell niche. Now, I've not listed this here, but when we're selecting a matrix, you've got to follow the protocol pretty closely. You need to think about things like thawing temperature, stability of that particular matrix, diluents, how long the matrix might last, right? All of which are really important factors that control the integrity of the matrix on which your cells are actually going to sit. Matrices have different initial coating concentrations, right? They oftentimes need to be optimized depending on the different cell line. And obviously your intended downstream application, but all important things to think about. You often, dilute your matrix of choice, use it to coat your culture vessel, often about, let's say, 1 to 2 mLs in the 6-well plate, for example. We would then often incubate those at the recommended temperature and time, and then ultimately they will be suitable for use for culturing PSC. So again, matrix is an important determinant for successful culture in feeder-free conditions, because it really does play a substantial role in a lot of important variables like cell growth, and it can, by very, very, very many means, influence things like pluripotency, confluency, cell identity, right? So important consideration. Other important considerations that we talked about, right, are media, right? And there's obviously lots of different systems out there for specific applications. But there's been some, as I alluded to before, some really obvious advances, I would say, in the so-called evolution of these different culture systems. Again, all the way from Jamie Thompson, right, in the late 90s with feeder cells and FBS, through to some other big leaps and bounds, as you see here on this slide, where, again, as I mentioned, the Thompson lab sort of re-examined the media landscape and tested newer combinations, sometimes with fewer components, to help us develop these more defined media, right? Giving rise to things like Essential 8, and then ultimately things like Essential 8 Flex and Stem Flex that we know nowadays not only support cells, but as the name implies in Flex, right, also allow for things like weak entry feeding. And a lot of that happens through stabilization of factors like FGF, which I'm going to touch on in a minute, as an important component of these feeder-free media systems. So this slide is just a quick side-by-side comparison of some of the components that comprise feeder-dependent and feeder-free media systems. Feeder-dependent media typically supports pluripotency and cell health using things like a DMEM-based medium nowadays, supplemented with factors like basic fibroblast growth factor, as well as serum, commonly nowadays knockout serum replacement, or KSR, which again is a more defined and I would say more reliable serum-free alternative that has been sort of modernized and optimized for PSC culture. These feeder-free conditions, these feeder-dependent cultures, right? They rely on those feeder cells as well. That's the big take-home on those. Again, and those provide a lot of those different growth factors and extracellular matrix proteins. And as we alluded to, these nowadays we see a lot more of these feeder-free media. I would say is probably a more popular choice for researchers, which again rely on the principle of omitting those feeder cells and then supplementing the culture components to compensate for things like nutrients, growth factors, extracellular matrix, right? And I've listed an example here of Essential 8 to support your PSC culture. So E8, just expanding on that example a little bit, is a nice media choice if you need a defined formulation. It has within it, as the name implies, only the required components for culturing PSCs. That leads to a lot less variability than some of the other, I would say, more complex media systems out there, because of a lot less interactions between these limited components and a much more simple defined substrate. We also remove BSA, which, as I said, can be a big culprit of variability. And I put a little bit of data along the bottom here, showing how in different batches or different levels of BSA in other media systems can actually lead to, as you see here in this graph at the bottom, quite a lot of variability in their capacity to actually support proliferation of pluripotent stem cells. So with our E8, we see a lot less variability, especially when we pair it with more defined matrices like vitronectin, for instance, where we see quite good improvements in PSC attachment, survival, high efficiency, etc. Routinely changing the media is something that if you've done cell culture long enough, we've all grown accustomed to. And changing the media is really, really an important consideration as well as far as the health of cells. There are still a lot of workflows that require daily media changes. But as I mentioned, there's some nowadays that are nice in that they allow you to skip a day here and there. Maybe not quite as absolute as this slide. I put daily and I put routinely underneath here, just to give you an idea that we are a bit more flexible nowadays, thankfully. Keep in mind too, as I alluded to before, that frequent handling of the cells that can occur with things like daily media changes can increase the likelihood of contamination. More hands-on time also can lead to potential increase in user error as well. So I think it's nice to have support in changing cell culture media, especially if you end up coming in a lot on the weekends or on holidays, for example. That can get old sometimes. We've all been there. But that need for daily media changes, and I would say a lot of consideration, if you will, in modern day design of more flexible media systems was based around FGF stability. FGF, or fibroblast growth factor, is quite a potent regulator of differentiation and proliferation in stem cells. And so we like to think of it as something that in many ways orchestrates the pluripotency process. But FGF is heat sensitive, right? So it has fairly poor stability at 37 degrees, meaning that we do quite need to quite regularly feed our cells to help maintain that pluripotency. And so loss of or fluctuations in FGF activity can, by all means, as you see here on this slide, lead to quite inconsistent culture conditions. This demonstrates here how much FGF levels can actually dip on a daily basis. And so what we did here, not me and Omar, this is some R&D folks at R&D who are much smarter than us. They developed this media formulation that basically eliminated the need for daily feeding by extending the activity of FGF and other key heat-sensitive components that are typically found in pluripotent stem cell medium. And what that does, as you see here in the blue, is really limits those fluctuations. This is a media essentially flex. As the name implies, it's a good degree of flexibility when you actually have to feed your cells. With this media and some of the other flex media, you can actually skip feeding for up to 2 or 3 days in a week, including 2 consecutive days. And we do this without compromising things like pluripotency and genetic stability, which is quite nice. This is just a graphic example of the capability of some of these flex media to improve FGF stability, as you see across the top here in blue, compared to some other media systems down below here. And then just to drive that notion home here, again, media choice is such an important consideration in stem cell workflow. At Thermo and most other companies, there's loads of different considerations and options for media choices. As you can see here, we've got a lot of different offerings that you can choose to help optimize your research, accounting for things like scalability, robustness, flexibility, even cell culture systems and media specific for parts of the workflow, right? I would say StemFlex in the middle here is probably our most well-rounded and versatile media. And again, we're also performing a lot of the work that we do nowadays in 3D culture rather than in 2D flat culture, right? As I alluded to. And so I think while we're on the topic of media, a good and important question to ask is, well, why should we actually consider 3D media or consider shifting to suspension or 3D culture over 2D or adherent culture? And I heard this in a conference once, probably about 10 years ago now or so, where the researcher that was giving this big sort of headline talk said, we can all agree that the world is, well, most of us can probably agree that the world is probably neither flat nor two-dimensional. So why on earth should our cell culture be, right? And that always stuck with me, right? These traditional cell culture systems, most of which we talked about today, again, they grow cells on these flat coated plastic dishes, right? But again, that's really not representative of what happens in the human body. And so nowadays, a lot of different cell lines can be adapted to spheroids and suspension culture, or even taken further out into organoids, which, again, generally speaking, helps drive a more predictive physiological response in these monolayer culture. Again, due to that sort of complex and multicellular nature of these cells in 3D, which better mimics in vivo biology. And as I keep alluding to, this is a much better bridge between, say, the research bench and the clinic. At Thermo, we have a nice media system for suspension culture called StemScale, which allows us to generate a large number of pluripotent stem cells. And again, not as a plug for that, but it's just a really nice and easy workflow to use. And we'll talk about suspension culture and media like StemScale a little bit further on down the road in another lecture. And then lastly here, along the lines of bench to bedside, right? This is a high-level overview of a 3D model, maybe in the setting of a drug development scenario. You can see in this picture here, we sort of illustrate, perhaps at a high level, a typical workflow of a drug going from the bench all the way through to a patient, hinting at things like the utility of 2D and 3D models, right? Both of which are very useful in these translational settings, especially as we're sort of making this stepwise progression to ultimately end up in clinical trials. We start here on the left with a screen of I don't know, a couple thousand or so compounds in a plate-based assay, for example. We take those positive or useful hits, we test those in a 2D monolayer culture to sort of test the effects of that media or that specific experiment in a living system, almost enabling the researcher, if you will, to fail early, right? And I would say fail more cheaply than if you'd taken it immediately all the way out to a 3D model or even a very expensive clinical trial. So this sort of in vitro testing helps us and allows us to evaluate different parameters like efficacy, different mechanisms of action, right? Functionality, even toxicity is super important as well, right? And then we take that data and we use that to test certain specific compounds, ultimately often in animal models and eventually often in human studies, we hope anyway. And again, all of these require a significant increase in cost and time as we kind of go from left to right here. And again, these 3D models are a nice bridge between things like lead discovery and in vivo studies because they recapitulate the human state in a much more physiologically relevant manner. Again, with all of that said, when we're thinking about what we need to grow cells in culture, we have to think about the cells themselves, right? Obviously, without them, none of this is possible. And again, I won't go into too much detail here, but these are a couple of basic characteristics that you want your cells to have in culture, right? These are telltale signs of happy and healthy cells that we want, right? This is a picture of essentialating vitronectin. You can appreciate here how tightly packed, I would say, these colonies are. They have nice looking, I would say, defined kind of refractive edges. Omar mentioned this last time, a high nucleus to cytoplasm ratio. That's important. This is something that helps control the S phase dynamics of the cell cycle. That synthesis phase of the cell cycle is when the DNA is replicated. So it's super important. A couple of considerations down the bottom here of how we actually take care of pluripotent stem cells. Very, very basic guidelines. But we have to think about how the cells are passaged, which I'll touch on in 1 second. How frequently we change the media, which we just talked about, to continue supporting cells. Visual inspection of the cells is really important. Here's some of these aforementioned featured. We also have to remove differentiated cells as well, right? And then, as we talked about with cell culture vessels, we have to plan accordingly, right? We need to make sure we have the dishes we need, the dishes that we need, or culture vessels that we need are prepared in advance, that they're maintained in a sterile environment, right? And we have to think ahead, right? Where are we going with our experiments? Because we have to anticipate and prepare for the next steps in whatever it is within your respective workflow. Again, keep in mind here that we are focusing for the most part in this course on feeder-free culture. And that I will say as well, a lot of feeder-dependent systems, there's nothing wrong with them whatsoever. They are still widely used, but they can actually be transferred quite easily to feeder-free systems as well. What we typically see is you'll see some residual feeders for a couple of passages before you can make that switch into feeder-free culture quite easily. There's also a number of different ways to inspect cells visually, right? We look for things like clumping, debris, even changes in media color are often indicative, right? And we can see things like this with the naked eye. Most labs, though, will have a microscope, I would say with probably at least a 5x objective. Most nowadays, modern microscopes have phase contrast capabilities as well. I'm sure you've seen a lot of these in publications or maybe even in the lab that you come from, but we also use immunofluorescence microscopy a lot to observe things like cell morphology and look at different markers that are important, right? And again, most functional labs will have these on hand. And then lastly here, as far as what's needed to grow PSCs, passaging is another important consideration. I'm sure many of you have a good amount of experience passaging cells, but it's important to consider especially the timing of when we passage, right? And if you look at this kind of graph over here on the right, this is typically based around a characteristic growth pattern of cultured cells, right? These are semi-logarithmic plots that show things like cell density versus the time spent in culture. You see here this sort of first phase, if you will, of growth after the culture is seeded. That's called the lag phase. It's a period that we know is relatively slow growth, all things considered, in which the cells are still kind of adapting, if you will, to that cell culture environment. The lag phase is typically followed by what's called the log phase here, or the logarithmic phase. This is where the cells proliferate exponentially. They quickly consume a lot of the nutrients in the growth medium. And then we know at the top here, when that growth medium, for example, is spent, or when maybe one of the different nutrients that we need is depleted, or at least running low, the cells kind of occupy all the available substrate as well. The cells will enter what's known as a stationary or plateau phase, in which proliferation is greatly reduced, or in some cases ceases entirely. So there's this kind of sweet spot for passaging is the take home here that I've marked here with an arrow on the graph. Usually that's around 75 to 80 or so, maybe 90% confluency in adherent cells. And again, this is just something to think about that helps keep those cells in optimal density for continued growth, and it also further stimulates proliferation, allowing you really to maximize the number of cells that you have to work with and keeping your cells much more healthy. Again, subculturing in suspension culture, we'll talk about this at another time. I would say it's less complicated than passaging in adherent cells. That's because the cells are essentially already floating in suspension culture. You don't have to detach them from the surface of the culture vessel. I would say making the process a little bit faster and sometimes less traumatic as well for the cells themselves. Just briefly, and as a point of reference here, I've listed a couple of tools for passaging adherent cells. Gentle shaking, scraping, enzymatic dissociation are all methods that are employed. Each have their different merits as far as different applications go. And there's loads of different types of dissociating reagents that you would employ for different cultures. And then lastly here, and switching gears just a little bit, these are just a couple of gentle reminders about some of the challenges and hurdles that you might encounter during cell culture. Again, I'm sure we've all had some nightmare stories of things that have happened in the lab. If you have one that's good or you'd like to share in the chat, please feel free to pop it in there. These are always interesting to hear. I've certainly had some. I know Omar certainly had some as well. I had a lady in my lab that refused to wear gloves until she was reported, and she did this for almost a year or so. And I swear she ruined so many people's experiments with that type of bad lab etiquette. But again, big picture here, keep an eye on things in the lab. Pay attention to your cells. Pay attention to your lab mates. Help your lab mates as well if they don't have the best lab etiquette or they're not working under the most ideal culture conditions. You've always got to be conscious of safety, of proper sterile techniques, because again, as we alluded to in the first lecture here, this can save you so much time and energy and stop you from wasting a lot of valuable resources by helping to prevent unwanted culture problems, contamination, for example, variability in the workflow, low cell viability, wacky looking data. So it's important to remain aware and understand all these different factors that can quite easily alter the outcome of your experiments. Again, going back to this slide, though, we really can leverage well-designed stem cell and disease modeling workflows when these pieces all fit nicely together, and you put the time and effort into doing so. Again, you'll end up with much more consistent, and I would say better and impactful data. And then last but not least here from me, I want to just very briefly take a couple of the principles that we talked about and apply them in the context of a quick case study. I think this one's a good one. Again, it's really quick, but what we did here is we employed cell line engineering to generate hypoimmunogenic hPSCs. This is some work that was done in collaboration with one of our longtime partners at UC San Francisco. She's really amazing researcher and clinician, Dr. Sonia Schrafer. If you've not heard of her or seen her speak, she's fantastic and has some really cool papers as well. So just to paint the picture in here, give a bit of background, what happens during pregnancy is the mother's immune system can actually develop pretty specific and quite localized tolerance to antigens from the father, paternal antigens, right, in order to help prevent rejection of the fetus. But one big potential downside, though, is that pregnancy can often induce systemic sensitization, right? Meaning that the mother can actually make long-lasting memory B cells that way down the road, years down the road, can later recognize and actually contribute to the complete rejection of foreign cells. Think of like an organ transplant, right? Especially, for example, those derived from that same child. And so quite interestingly here, what Dr. Schrafer did and her team was address this issue by generating this hypoimmunogenic derivative of pluripotent stem cells, one that actually enabled them to evade immune rejection upon transplantation into actual immunocompetent recipients, right? So they looked at gene expression first and foremost of the syncytial trophoblast. That's the cell type directly at the maternal fetal interface. And what they postulated was that these hypoimmunogenic hPSCs could actually be generated through mimicry of those different gene expression patterns found at that placental barrier, right? The barrier between the maternal and the fetal blood. So more specifically, what they looked at here was inactivation of MHC class 1 and class 2 target genes and overexpression of CD47, in which they hypothesized that this would allow the cells to avoid recognition by the immune system. So you can think of CD47 kind of as this don't eat me signal here, preventing macrophages and other immune cells from engulfing and destroying those healthy cells. So what they did is they took those iPSCs and then differentiated them into different cell types, endothelial cells, cardiomyocytes, smooth muscle cells, and then transplanted those into recipient mice in this case in order to assess survival and different immune interactions. And again, just in line with some of the content that we've discussed in this course so far, from a genome editing perspective, they performed a knockout in the human and mouse iPSCs using CRISPR-Cas9. They looked at two different genes that we know are associated. They're listed here with class 1 and class 2 MHC, which were then simultaneously targeted for CRISPR-Cas9 mediated disruption. They then made a transgenic line, right, of that double knockout, again with CD47. Not to get too far into the weeds, but they used a lentiviral approach here, right? So those edited iPSCs were transduced with a lentivirus carrying CD47 to ensure overexpression. And then they differentiated those into cardiomyocytes and endothelial-like cells, for example. But obviously the million-dollar question here was whether or not this was actually going to work. And so just a very brief snippet of one of the figures from this paper in Nature Biotechnology. They demonstrate here successful generation of these hypoimmunogenic hPSCs and their differentiation into immune-evasive cell types. In this case, they used luciferase tagged cells in these kind of humanized mismatched mouse models. And they found that while both initially here, as you see on the left, the wild type and these engineered cardiomyocytes initially invaded rejection, over time, only these hypoimmunogenic cells survived long-term without giving them additional immunosuppression, with the wild type cells being cleared in about 20 days or so, whereas these hypoimmunogenic cardiomyocytes stayed with a much stronger signal and stuck around for a lot longer, about 50 days or so. And again, all of this was published in Nature Biotechnology, if you'd like to see more. So again, in all, they used a CRISPR-Cas9 system and lentiviral vectors. They engineered these iPSCs. They differentiated them into terminally differentiated cells. And they showed that these derivatives were able to actually evade immune rejection and survive long term. Again, in the big picture here, I think showing quite significant potential for things like transplantation therapy, and sort of setting the stage for a very interesting paper and quite a promising study. That's it for the first part of the talk here. Again, I put a quick overview slide here just as a point of reference for you. We will share these slides after the talk, and I've also put some useful references and resources down here at the bottom, including a link to today's case study if you want to dig a bit deeper. Again, I'll stop here in the interest of time, though, and I will hand the reins over to Dr. Min-Fen Ho from the Mayo Clinic to talk about her exciting research. And Kitai, if you want to say anything or a few words to introduce, feel free to do so as well. Thank you.